Well, good morning, everyone. I think we'll go ahead and get started. Welcome to the March edition of the Spatialomics Interest Group here at the University of Minnesota. Somehow it's March already, but welcome. We're going to follow our typical meeting format today. Please do mute yourself unless you have a question. Feel free to raise your hand. Feel free to drop a question in the chat box. We will have a discussion and QA at the end of today's talk. Our next meeting is Tuesday, April 25th. We will have a SIG member presentation, so that's exciting. And I guess without further ado then, we're excited to have our spatial partners 10x Genomics here to give a talk about what's new there. I'll go ahead and introduce Eve Marie, who is our local rep here at the UMN and beyond, to introduce our speaker. So Eve Marie, thank you very much for being here today, and I will turn it over to you.
Thank you so much, Grant. Well, good morning, everyone. Thank you so much for attending this 10x Genomics presentation, where we're going to give you some updates on our spatial transcriptomic products. Without further ado, our speaker today is Leilani Marty-Santos. She's our Spatial Science and Technology Advisor. So thank you again, everybody. And please feel free to put any question in the chat, as Grant mentioned. Thank you.
Hi, everyone. Thank you so much for being here with us today and for allowing me to speak to you in this meeting. I was mentioning to Grant earlier that I've attended a couple of these, even when I was a postdoc, and it's one of those things that I really enjoy this group. I know that you always have wonderful questions, and so I encourage you to ask those in the chat throughout, or as Grant mentioned, asking them at the end. But yeah, today I'll be speaking to you about our spatial platforms at 10x. I think most people that know us from our single-cell biology, so our Chromium single-cell platform, which is the single-cell biology platform of choice for a lot of users. We've enabled a lot of our researchers to be able to work with many different things. So from whole transcriptome single-cell sequencing to working with things like ATAC, epigenomics, and other aspects as well in single-cell with a bunch of different modalities. And so I think people know us very well from that. Along with our Chromium single-cell platform, we also have our Visium spatial platform. And so this falls into what we call our hypothesis generating technologies. Both of these are whole transcriptome based. And so because of that, they're more of discovery platforms than our technologies. The other thing about these two technologies is that they're NGS based. And so you're working with that sequencing component to be able to get to that whole transcriptome information. By contrast, we just launched our Xenium in situ platform, which I'll be talking to you very briefly at the end of my presentation today. And this is going to be what we call our hypothesis-based testing or focus modality. It's going to allow for really high resolution views of your transcript. It's going to be a much more focused panel, so say about 300 genes currently with our recent launch, and it's going to be an integrated microscopy platform. However, I will be focusing most on our Visium platform today. So as this animation plays, I do want to highlight that this is what a lot of people come to us for. So they're probably used to working with that single cell biology component and working with the analysis in either UMAP or Tisney plots, and that may be something you're very familiar with visualizing. However, you can see as this animation moves back and forth, the ability to look at that gene expression and put it into the tissue context. By being able to do this, it's going to allow you to be able to do further analysis, so understanding where that expression is happening, what cells are having that expression, what neighbors are interacting with each other, and how does that change expression patterns overall. Those are just a handful of questions that we have a lot of biologists looking at, and I think it's going to be important for a lot of our researchers to have that further granular information. So not just the single cell biology, but really understanding that spatial context overall. So for our Visium spatial gene expression technologies, we tend to talk about our two different chemistries. So anyone who has worked with our direct placement technology, which is the first technologies that we've launched for Visium, may have come across these. So for FFPE tissues, anything that's going to be a fixed modality, one of the problems that you run into when you're doing this for your histology is that when you're working with RNA, that formalin or that PFA can usually cause some level of degradation to your RNA. especially this tends to happen towards the poly A tail, so that limits that traditional poly A capture methodology that a lot of our single cell assays use, for example. And so to circumvent this, we've actually created whole transcriptome probe sets. I'm showing here, for example, that we have probes that target about 18,000 genes in the human, 20,000 genes in the mouse, but it still gets you to that whole transcriptome information across the whole tissue section, because this way it allows us to target those specific genes instead of looking for a poly capture methodology that may not be there. For fresh frozen tissues, this is a poly A capture methodology. And because of this, you're actually able to work with a lot of different species as long as you have a reference genome to align to. We call this our species agnostic technology. So for a lot of our users that may be working with those so-called non-traditional model organisms that aren't human or mouse, this is going to allow them to work with those tissues of interest. The basis of our Visium spatial technology is going to be the spatial gene expression slide that you're seeing here. And so what you can see here is our original direct placement technology. As its name implies, you would be directly placing your tissues on one of these 4 capture areas that you're seeing here on the left-hand side. Each of those capture areas has this fiducial frame shown in red in this cartoon, and that's going to allow for the alignment of your image. So as you place your tissue on there, you know what orientation it will have. You can move this through an imaging process software, and even if your image flips, you know where each of your components of your tissue was located. Within that fiducial frame, we have this area that is 6.5 millimeters by 6.5 millimeters and has about 5,000 barcoded spots. Each of those spots is going to be about 55 microns in diameter, and it's going to contain millions and millions of these capture oligos that I'm showing in cartoon format here on the right-hand side. So what you're seeing here with this oligo is that you'll have a partial true-seq adapter. As its name implies, this is going to allow for sequencing. You then have a spatial barcode. And the way that I usually explain this is that it's going to be the equivalent of an X and Y coordinate on that slide. You can see from this cartoon how this spatial barcode is color-coded green, that has a specific sequence that aligns it with this spot here, and all the way back to this specific position on the slide. And that's how you know where you're gathering your transcript from your tissue overall as you take that image and they later align it. The next component of our oligo is going to be a unique molecular identifier. As its name implies, it allows you to identify each of your unique capture targets. And so when you're working with our poly-A-based technology for fresh frozen, you're capturing and quantifying directly the mRNA readouts. If you're working with our probe-based technologies, then you're going to be quantifying how many probes you've captured. And lastly, you have this poly-DT. For our probe-based technologies, we have a synthetic poly-A tail that can be bound to this poly-DT, and that's what allows for the capture on the slide of those probe targets. When you're working with fresh frozen, you're going to be allowing for the capture of that poly-A-based mRNA substrate. This is going to be a simplified diagram of the workflow. In true 10x fashion, anything that you need will come with our kits. And so what you're going to start with is doing your tissue prep. So if you're working with either fresh frozen tissue, for example, you would be sectioning your tissue on a cryostat, placing it on the slide, doing some OCP washes, and then a permeabilization step that will allow you to release RNA. You're also going to be doing a tissue staining component. So that's going to allow you to do your imaging. You would then take and save that image and move from there into barcoding and library construction. It's going to be a very similar process for those of you working with FFPE, where you would do your standard traditional deparaffинization, your staining, and then go into the barcoding and library construction process as well. Once you've done that barcoding and library construction, which I'm sure Fernanda and a lot of the cores, the people that work in the cores there can help you with, from there you would then submit for sequencing. Once you get those sequencing files back, so your FASTQ files, you would then run this through our Space Ranger pipeline. And what that Space Ranger pipeline does is it takes that image that you took in the second step of this workflow and that sequence information, and using that spatial barcode that I mentioned earlier, it's going to align specifically where each transcript was on that capture area of the slide. Then from there, you have 2 choices for your visualization. You can either use our Loop Browser software, which I'll show in a second. That's a free software that you can download today, and we have a broad variety of different sample data sets that you can look at using that Loop Browser software. The other choice that users may opt for is going to be using a third-party community developed tool, so something such as Seurat or CTD or other pipelines. One of the advantages of 10x Genomics is that none of our data is ever locked into any particular format, so you're really able to work with it in whatever way you find best for your research. This is an example of that loop browser software that I mentioned earlier. And so what you're seeing here on the left-hand side is the ability to do your whole transcriptome information overlay on that whole tissue section. We're zooming in pretty far, but what you're seeing here is going to be that gene differential expression across the tissue. So as you see, once again, these spots being pulled up, you can imagine that the genes that are being expressed within those red spots that are coming up are going to be different than the blue spots that are adjacent to it. And this is just based on principal component analysis. On the right-hand side, you can see the ability to select an area of tissue. You do not have to do this a priori. This is something that you will always have your whole section, whole transcriptome information. But I know for some of our users, as in the example given here, which is a breast ductal carcinoma in situ, you may be interested in choosing a tumor region, for example, and then comparing that to, say, a normal margin area, and be able to then identify what's going to be the gene expression changes throughout those two specific regions. And this is something that you can analyze as simply as taking this out, exporting it to Excel from Loop Browser, and being able to just look at levels of those transcripts. The next part of my talk is going to focus very briefly on a couple of different case studies in terms of what people have used Visium for. I know that I have a list up here of a couple of different fields of interest that people have used Visium, but honestly, the sky is the limit. We have so many people using this across different modalities, and I want to make sure that people understand that this is going to be something that you can use across the board. The first example that I'm showing here was actually done by our internal R&D team. And so this is looking at this amyloid precursor protein overexpressing model. And so what this is going to look at is a model of familial Alzheimer's disease. And so in this case, this overexpressing or transgenic model is showing this particular high levels of gene expression that consistently came up whenever we were comparing it to the wild type mice. The gene that we realized was really highly overexpressed compared to other genes that we were looking at was this promelanin concentrating hormone. When you think about promelanin concentrating hormone and its function in neurobiology or neuroscience, it's one of those genes that's going to be more associated with regulation of the sleep-wake cycle. It's also associated with impaired cognitive function. And so it's really interesting to see this when we put it in our Visium technology. And so what you're seeing on the right-hand side is going to be at the top right panel, comparison between the wild type expression and the transgenic expression of that promelanin concentrating hormone. You can see here is going to be that this gene is normally only expressed in the hypothalamus. Once again, something that's very interesting when you think of the regulation of the sleep-wake cycles. And it's really not expressed in any other place in the brain. However, when you look at the transgenic model, you can really see how highly this is overexpressed compared to that wild type expression pattern. You can also see how this is expressed across the brain overall. And so when you're looking at something like this, you can understand why you have that impairment of cognitive function if you're affecting cortical layers, for example, or other areas of the brain, and how that sleep-wake cycle is going to be dysregulated when you have this overexpression that is much higher overall. The beauty of our Visium technology is because it's whole transcriptome, you're really going to be able to look at your whole gene approach in that tissue overall. And so instead of using something that may not be as relevant to your research, let's say, the common beta-actin loading control that many people use when they're doing either their qPCRs or whether they're doing protein assays, you can see here the ability to look at genes that are relevant of interest. In this case, we're showing neurogranin. This is going to be a pan-neuronal gene that expressed across the brain with its expression most highly being expressed within the cortical layers and within the hippocampus. And so you can see how this expression pattern is actually very much the same between the wild type and the transgenic. So you can see how even genes of interest can also serve as internal controls. And this way you can see what is truly changing and what is not across the entire transcriptome and across your entire tissue. The next example that I want to give was done by Joachim Lohnberg's group. So in this case, what they were doing was using spatial maps of prostate cancer samples. So they're taking and sectioning prostate samples overall, doing H&E staining, submitting these for blind annotation by pathologists, and then taking that information and doing our Visium workflow using that Visium workflow, they then went through and kind of designed these factor analysis, so these factor signature or factor activity maps, being able to determine what gene signatures or what gene factors are associated with the prostate cancer progression. And so what you're seeing here on this top panel is a good correlation between the pathologist annotation and those factor activity maps associated with different areas of the tissue or the cancer progression overall. So this blue arrow here is highlighting this area that was identified as being cancerous, with particular emphasis on this red oval here. You also have these prostatic intraepithelial neoplasias, so kind of an early stage where you're beginning to see changes in that morphological tissue at that point. When we look at the factor activity maps, we can see how nicely these correlate with those pathologist annotations. So we see that cancer signature in the same region that the pathologist annotated, same with those prostatic intraepithelial neoplasias, and then everything else has been identified as stroma. However, one of the things that they were able to find with their publication is that sometimes we have these transcriptional changes that predate any visible morphological changes. And so when this pathologist went through and did their sample annotation, you can see in this case, they had not identified anything as being abnormal in this particular sample. However, we were able to use the Visium workflow to then use factor activities and map this region that hadn't been identified as being cancerous based on those signatures. And so this really highlights the fact that sometimes having that whole transcriptome information, that whole tissue section can really lead to discoveries that are going to be important in understanding the biology of the tissue, even if we can't see those changes in a visual level. This was actually our first FFPE publication, and so this came out last year. And so in this case, what we're seeing is looking at tumor immune biology. And so in this case, it's looking at these tertiary lymphoid structures in a renal cell carcinoma. And the reason this is going to be important is because these tertiary lymphoid structures are actually these hotspots where B cells begin to differentiate and become these anti-tumoral antibody production hotspots within the tissue overall. We have this factory that is going to be producing this anti-tumoral response. So what you can see here on the left-hand side is going to be this tertiary lymphoid structure that was identified in that H&E image that was then used to correlate the data that we are seeing on the right-hand side. They took an adjacent section, performed some CD20 staining, and so what you're seeing here is going to be that location of those B cells in the tissue. And then they created, similar to the previous publication that I showed, this factor activity map for those tertiary lymphoid structures. So what signatures can be associated with them and where are they in the tissue? And you can see how nicely this correlates with that pathology annotation. Then they went through and were able to look at many different gene signatures overall. So you can see this naive B cell signature and how they kind of surround that tertiary lymphoid structure as they begin to migrate in. And then you can see as these cells begin to differentiate, where they begin to locate specifically within that tertiary lymphoid structure. So you can see now as these B cells are differentiated into plasmablasts and ultimately into those plasma cells that I mentioned are those factories for creating those anti-tumoral antibodies are located within that tertiary lymphoid structure. You can then correlate this with the actual antibody production itself within that region. And you can also visualize the COL1A1, which is one of the extracellular matrix genes and proteins that is associated with providing that niche, that structure for those tertiary lymphoid structures. So really the ability to get down to the biology of what is happening at different time points, so to speak, in the differentiation of these cells and what is happening in the tissue biology itself. Lastly, I want to show the ability to combine and get down to even more granular data when you're working with both of our whole transcriptome platforms, both our chromium single cell biology and our Visium platform. This is a publication that came out from Holger Haynes' group in Barcelona, and they decided to undertake this massive tumor immune microenvironment atlas. And so what they did was they analyzed 217 different patients presenting with 13 different cancer types, and they took 500,000 single cells. From there, after doing that initial analysis, they were able to really look at and determine what proportion of these immune cells are normally present in different cancer types. The idea behind this is if we understand what the immune biology is behind tumor progression and what immune cells are there, we can use these immune cells as targets for precision oncology. They then took this one step further by combining it with Visium. In this case, they used a community developed tool called Spotlight to integrate the data. And so what I'm not showing here is the underlying H&E image, but you can see how they annotated this breast ductal carcinoma in situ sample, where you can see these fibrotic regions that are characterized by expression of extracellular matrix genes. This is pretty normal when you're looking at these different breast cancer samples, where there tends to be this fibrotic encapsulation of the tumor sample itself. And then you also have these hormonal status regions. So looking at, for example, this region where there is HER2 expression by itself, and then this other region here where you have HER2 and estrogen receptor 1. They then overlaid the immune cell data that they had obtained with single cell and looked at the different locations of those immune cells overall. So if we look at that HER2 estrogen receptor 1 double positive region, you can see, for example, how much this area is enriched for these memory CD4 T cells overall. And then there's some expression of these CD8 pre-exhausted T cells as well in this region. Now, I am not a cancer biologist by any means, but one of the things that struck me when looking at this image is that you also see those pre-exhausted CD8 T cells in that HER2 positive region. And for me, it's an interesting correlation that might be indicative of function. Maybe those CD8 T cells like to localize specifically with those HER2 positive tumor regions. And so this is something that could not have been determined if you're doing a dissociation of the whole tissue sample. Putting it into the spatial context will really allow you to explore the biology and the interaction between these cell types overall. Because we have a lot of users that are very interested in doing this type of work where they're integrating that single cell and spatial data sets, we've actually added these data integration analyses guides to our website. You have the ability to go through, see tutorials and different communications that we have working with common tools such as RCTD, Seurat, self-location, and others. And for anything that I'm presenting here, I do want to highlight that if you have questions, feel free to ask them at the end, drop them in the chat, or feel free to reach out to your 10X team, and we're happy to send these references along as well. Now, the next part of my talk is really going to focus on the advancements that we've made with our Visium technology. And the first one of these came out for the latter half of last year. And so this is going to be the ability to bridge the worlds of histology and genomics. And the reason I'm saying this is when we work with our Visium workflows, there's usually two groups of users that we have. So we have the people that are very comfortable with histology, that are very used to doing imaging and sectioning workflows, but maybe they don't have that NGS library construction experience. On the other side, we have a lot of those users, as I mentioned, that may come to us from that single cell biology side, where they're incredibly comfortable with working with the molecular biology aspect, but perhaps aren't as comfortable working with the histology portion. And because of this, we've created our CytAssist instrument. What our CytAssist instrument does is it allows our users to get more from their samples. And what I mean by this is that it enables our users to work with expanded sample formats. So you can start with, instead of that Visium capture slide that I showed at the very beginning, you can now start with a plain glass slide. And so one of the advantages that this would have is going to be allowing for the simplicity of just sectioning onto a plain glass slide, or maybe a charge slide if you're working with any tricky tissue types that may need further adhesion. It also enables our users to be able to work with samples from collaborators. So maybe you're working with a collaborator at a different institution and they can't ship you a block that you can section from, but you can get them to send you a couple of slides. Same works with a lot of biorepositories where they're not able to necessarily give you their full blocks, but they're able to send you a few slides of your sample of interest. And then one of the other sample types that we've added that I'll mention very briefly is looking at archival samples as well. So something that you may already have sections, stain, and cover slips sitting in your laboratory might be eligible for this technology now. So you have that advantage of working with a sample that you never thought you could use again. This workflow is still going to be compatible with either our H&E or immunofluorescent staining. So you will be able to do those on that same tissue section, do the visualization, and then do your downstream workflow. So you're really able to see all that whole transcript information from that same tissue section. And then you have the ability to go through and choose your best tissue section. Maybe you're working with a rare cell population. And so being able to section onto plain glass slides, doing that staining ahead of time, and choosing which slide to work with instead of having to section fresh from a block and hope that you're the perfect region of your tissue is going to be the better option for you. Lastly, one of the advantages that we've added with our site assist is going to be the ability to work with additional capture size areas. So this blue square here that you're seeing outlined on this tissue is our original 6.5 by 6.5 millimeter capture area that's available for direct placement. It's also available on the site assist, but we've additionally added this 11 millimeter by 11 millimeter capture area as well. So in terms of working with our site assist, as I mentioned, your input is going to be a little bit different. You're going to be working with 2 tissue slides. So we're working with plain glass slides. So these could be those usual superfrost slides that you may have in your laboratory that seem to be very common across histology laboratories, or any slide of interest. From there, you would do your traditional histology workflows. So you're going to do your H&E staining or your immunofluorescence staining, take your image, and then you're going to do a probe hybridization overnight. After you've done that part of the workflow, you will then take a reconfigured Visium slide that has these 2 capture areas now. You're going to place both of those into the instrument. So what you're seeing at the top of this instrument is going to be these 2 slides that have these stained tissue sections on them. And then at the bottom, you can see that newly reconfigured Visium slide that has these reagent chambers. As you go through the workflow, you'll add some reagents to those reagent chambers, and then you're going to close the instrument, bringing those tissues into close proximity. I very unscientifically explained this as being similar to a panini press, but that seems to help with visualization. And so as you close that instrument, you can imagine you're bringing those tissues to close contact with that Visium slide. And so what you're seeing here at the bottom is going to be the explanation or a cartoon graphic where you can see how those probes that were hybridized onto that plain glass slide are going to then be transferred over to that Visium slide that still has those capture oligos that I described earlier. After you work with the site assist instrument, you then are able to look at the Visium slide with those oligos that have been captured on them. So you have now these probes that have been transferred over. And so at this point, you can use that for your library construction. And then you also have an additional set of images that is generated by the site assist. So this has a camera here underneath. And so you can take your original images that come from the plain glass slide that did not have the fiducial frame. You can now have that specific orientation and understand what specifically was transferred in what orientation and perform your alignment using both of these images. This is a simplified version of the workflow. So you already saw something similar to this previously with our Visium direct placement technology. Here you can see, you're starting with that plain glass slide or standard glass slide. It's showing FFP, but this is also compatible with fresh frozen tissues. It is going to be limited to human or mouse for now, just because we are working with a probe-based approach. If you have questions about that, feel free to reach out to me. I'm happy to talk to you about work that people are doing using these probe sets potentially on non-human primates or other organisms, but we can talk about that afterwards. And then from there, after you do your sample preparation, as I mentioned, you would do your traditional histology workflow, take your pictures, and then that overnight probe hybridization. After that, you place your slides on the instrument, you're going to do that transfer, and then from there, it's very similar to the original workflow, where you're going to do your library construction, your sequencing, and then use our Space Ranger pipeline to do your analysis afterwards. I really want to show how the power of having this additional capture area size is going to be important for anyone that is working with a large tissue sample. So what I'm showing here is going to be the 6.5 by 6.5 millimeter capture area containing these human breast cancer 4 biopsies. And while you can see that the biology is excellent, you can really see these nice clusters that are created in terms of gene expression profiling. Now you can see just how much of that biology may have been missed if you're working with a small capture area on a large tissue size. There's some cluster patterns you can see here that weren't identified within the small tissue section, and it really gets you much further in your biology when you're trying to answer questions about what is happening within the tissue overall. I mentioned earlier that we've really made this a tool for our enabling of our users. And so what you're seeing here is a 17-day mouse embryo section. Hopefully that gives some people context into how large this tissue section is. And so you can see how the different tissue slides, so 2 plain glass slides containing serial sections of this tissue, were placed on the instrument head. And so we can see here how we're aligning for either the mouse embryo head or the mouse embryo torso, specifically using our guide. So these blue lines here, which are colored just for visualization purposes, they would be a thick, dark gray bar on our instrument for use capabilities, but you can see how you can align to these thick bars for our 6.5 area, and then you have this guideline at the corners for the 11 by 11. So you can see how you can choose specifically these areas of interest whenever you're working with a 6.5 or the 11 millimeter by 11 millimeter capture area, if that's what you're interested in. And this is an example of the data that's been generated from it. So once again, you have that whole transcriptome gene expression profile here on the left-hand side. So looking at those gene differential expression patterns across different tissue types. And then you have the gene expression of specific genes of interest. So looking at those levels overall, so whether it's something that's highly expressed in the eye or something that would only be highly expressed in the extremities, but being able to do that visualization of individual genes of interest as well using our Loop browser software, for example. Lastly, in terms of the things that I wanted to talk about as came out with our release for the CytAssist late last year was the ability to work with archival samples. So I mentioned earlier that we now have the ability to look at samples that have been sectioned, stained, and sitting in the lab for a while. This is an example of that. So looking at this human lymph node sample, which has been H&E stained, hard mounted, cover slipped, and stored for 5 months at room temperature within the laboratory. From there, it was decover slipped and then run through our Visium CytAssist assay. You can see how nicely these gene expression clusters mirror the biology from that H&E stained sample. You can even see this with a specific gene of interest, in this case, looking at RGS13, which is going to be associated with those B cells in the germinal centers. Once again, you can see just how beautifully this mirrors that tissue biology overall. And I know that this is not necessarily the oldest sample that some people may have in their laboratories, but as I mentioned, we just launched this technology late last year. And so I currently have users that are really testing the limit of the biology. And so they're really going into much older samples. I do want to highlight that one of the things that we found, it's not so much the age of the sample itself, but how well that sample was treating during its fixation, its processing, and its storage conditions afterwards. We do have QC metrics that will allow you to proceed or not with a sample of interest. So if you're interested in this protocol, please reach out. The next thing that I want to talk about were some key announcements that we made recently at our AGBT meeting. And so these include expanding sample access, working with additional analytes and multi-omics, and a higher resolution system that will be only coming out for the CytAssist. So I wanted to show here how our CytAssist has been demonstrated to outperform direct placement methodologies. So whether you're working with fresh frozen, which is what I'm showing here, or if you're working with our FFP technologies, we found that you're really able to get more enhanced coverage of your data when you're working with our CytAssist instrument. And so you can see the comparisons between serial sections that were worked with on either our CytAssist instrument or our direct placement methodology, and to see how many more clusters, for example, you can see in this core biopsy sample, and just how much you can see individual genes overall as well with our tissue biology. One of the other things that we found is that we are actually able to drop our caveats in terms of RNA quality when you're working with our samples. Our normal recommendations have always been looking at the RIN score of 7 or above for our fresh frozen technologies. Because we're using this probe-based approach, and because there's some changes into how the generation is being done of the RNA capture methodology, we have been able to see that even with these really good RNA quality samples of 7, you have this enhanced detection using the CytAssist of either UMIs or genes per spot. And we are actually able to reduce these standards even further for our CytAssist technology. So we found that samples that may have been thought to have questionable RNA quality previously, so samples that may have an RNA RIN score of 4 or above, are now compatible with our workflows. And we're able to generate beautiful data with these samples, even if they had that spotty RNA quality overall. And we've seen the same thing for our FFP tissues. Our prior recommendation was a DV200 of 50% for our direct placement technology. We've now been able to relax those standards to 30%. And this is not a steep cliff. I know that we have users that, because they may have a single patient sample that they really want to analyze, they'll sometimes work with that, even if it's potentially an unsupported protocol, and they've been able to generate beautiful data with RIN or DV200 scores that are less than what we recommend. The other sample compatibility that we've added is working with tissue microarrays. And so you can see this alternate microarray here where we're alternating kidney and brain samples, and you're really able to go through and identify each of those individual samples as being individual tissues when you perform your analysis. So there's no confusion between those spots or any tissues overall. And you can really see how even with these tiny microarray cores, you do have some spatiality to your biology. So you can see how you have these different clustered regions across the tissue samples that are present. The next thing that I want to talk about is our upcoming multi-omic solutions. So being able to do what we call spatial proteogenomics. And so in this case, you will have the ability to get 3 different modalities from a single FFPE tissue section. So you have here the example of a tonsil section that was stained with H&E. Same tissue section was worked with doing the whole transcriptome approach. And on top of that, on that same tissue section during the same run, we were able to add a 35 plex that's going to be an antibody panel looking at immune profiling. And so all of this was done at the same tissue section at the same time. And you can see how you're really able to see these discrete clusters when you're looking at different markers of the tissue, both for protein and RNA modalities. And so this is coming soon, and we're really excited about it. Last thing that I want to talk about with advancements coming for Visium, specifically all of these approaches are for their CytAssist instrument, is going to be our Visium HD. We know that we have different researchers that are working with very complicated biology, and sometimes that biology requires higher resolution. While a lot of our users have been able to get down to single cell level information by doing data integration with single cell biology in our current Visium approach, we know that some users want to be able to do this from the get-go with our Visium platform. So we've heard them, we've been working on this for a while now, and so we have our Visium HD release, which is upcoming. So what I'm showing here is this FFPE mouse intestine. I do want to show these transcripts that were generated, or the transcript readout that was generated using that Visium HD pattern. And then I want to call out that we're really focusing on this area here. So when we look at that specific area, this is the H&E stain, and this is CytAssist data. I want to point out this is not immunofluorescence. This is going to be those individual transcripts that we're looking at that single cell scale, so sub 10 microns. And so it really highlights the ability to look at the transcripts overall, look at these individual cell types that are identified based on that marker gene expression. And it's really going to open up the biology for our users as we move forward with this Visium HD technology. So hopefully I've impressed upon you these important upcoming developments for Visium. They are only going to be available on CytAssist, but we found that the CytAssist is really going to enable our users to have better gene sensitivity and specificity. It allows for more tissue compatibility. So not only are you working with plain glass slides as your starting point, but now you can work with things such as tissue microarrays, FFP, or fresh frozen tissue across the board, and be able to get a lot more information. And the last thing that I want to talk about very briefly is our upcoming Xenium in situ platform. And while I say upcoming, we've already begun shipping these instruments. And so we started shipping them in January, and we've already seen users being able to generate their data. So this is going to be a subcellular in situ platform that we're going to be releasing soon. We're going to have this biology overall where we're looking at this industry subcellular platform. And so I know that we have a lot of users that are really excited about this release coming from a company that they trust, such as Genomics. So when you work with Xenium, it's going to be very similar to those histology workflows that you're already familiar with and may be familiar with from Visium. I would say it's much easier even than Visium overall. So you're starting with sectioning tissue onto a 12 by 24 millimeter capture area. So you can see here how, for example, someone has really loaded up that tissue area with 3 samples overall. From there, you would do a DNA probe hybridization that's going to target RNA. I'm showing the example of one individual probe here, but our starting point currently is 300 genes and panels that are off the shelf and completely validated. but you also have the ability to work with these panels that are going to be customizable. So you can either add 100 genes of interest or fully custom panels as well. From there, we do a highly specific ligation. And when I say highly specific, we're talking down to the single nucleotide level. We already have publications from the prior form of this technology where we've looked at isoform specificity. And we recently showed some proof of concept data at AGBT where we were able to look at single nucleotide variants or single nucleotide polymorphisms. So that shows just how specific our technology is. Then from there, after you do that highly specific ligation, you would then do a rolling circle amplification. And what this is going to do is it's going to generate multiple copies of this gene-specific barcode. The reason that's important is because you then have the ability to place those slides onto the instrument. And what the instrument will do is it'll wash on fluorescent oligos that will bind to those gene-specific barcodes in iterative cycles. So what will happen is you'll have some fluors washed on by the fluidics component of the instrument. It will move to the right-hand side, generate an image, and then come back and wash those fluors off and be able to do this in iterative cycles. Being able to do this is going to allow us to reach those multiple genes of interest and create individual optical signatures for each of those genes, ultimately allowing us to have a very high level of precision down to the single transcript level. Xenium is going to leave your tissue morphology intact, and what this means is a non-destructive methodology. So you have access to that same tissue post-run on the Xenium instrument. So if you're interested in doing an H&E staining for the traditional pathology view, doing immunofluorescence for added multi-omics capability, and I will mention that we're also adding protein to the system coming soon. You have the ability to do those visualizations as well. And one of the things that we've shown in-house is the ability to actually do Visium post-Xenium. So you would be able to do Xenium first, have that really high resolution image of your individual transcripts, but maybe in a more focused panel. So maybe looking at those 300 genes, as I mentioned. But then you can take that Xenium slide, put it at the top of the cytosys instrument, transfer the information over to a Visium slide, and then be able to generate whole transcriptome information from the same tissue section. So once again, really opening up avenues in terms of discovery and the biology that you'll be working with. We have a lot of available off-the-shelf panels currently. We have users that are already working with our human breast and mouse brain panels. We have a lot of panels that will be coming up, including multi-tissue and fully custom panels. So if you have questions about this, please feel free to ask. And then the last thing that I want to talk about very briefly is our Xenium Explorer software. The idea behind the software, which is already downloadable with sample data sets for anyone that's interested, is that it's going to be as easy to use as something like Google Maps. And so I'll show this here where you can see how easily you can begin looking through and panning through tissues. You can see how you're able to zoom in very easily. And so what will come up here as we begin to move are going to be these cellular outlines that are designated based on our nuclear segmentation algorithms. And so this is going to allow us to visualize each individual cell. This is going to be important when you want to align, for example, different transcripts. So what you're seeing here are groups of different transcripts identified by cell type based on each individual color. Each individual symbol is an individual transcript in and of itself. So for example, as we go through this animation again, you'll see these different blue colored transcripts that are identified with particular cell types. And in this case here, you have T cell transcripts that are identified in red, B cell transcripts that are identified in pink, and you can go through and be able to do the visualization overall. Here you have the ability to look at our density map. So looking at cellular neighborhoods, understanding the interactions overall, or even just the clusters that are identified based on the transcript expression patterns as we go through. And then lastly, because sometimes simple is better, you can use our lasso tool, for example, to do something as a simple area selection. Maybe you just want to compare two regions of the tissue. You can do something as simple as exporting it out to Excel. Maybe you want to create a bar graph looking at the expression patterns of particular transcripts of interest overall. So that's our Xenium platform. I'm going to leave this up for a couple of seconds, just if anyone is interested in using that QR code to get to our interactive data sets and information about Xenium in situ. Once again, you have Eve Marie and myself who are your specialist for the area, and so we're always happy to help with any information. Your whole 10X team is always going to be happy to help. And then the last thing I want to focus on is that we know that all of our technologies interact well with each other. We recently put out a preprint that you can find using this QR code, where we actually use serial sections from the same block to do an FFP tissue association and get down to single cell information. This is something that wasn't an ability that people had to do just 6 months ago. We've used the same section for analysis with our Visium spatial platform using our CytAssist and then Xenium in situ to get to really high level information about our about this tissue biology overall. So this gives you the information about that entire toolkit. And so with this, I think I'll stop and take time for questions.
Before we take the question, Leilani, I would
Oh,
like
sorry.
to see, oh, no, you're totally fine. Alex, would you be okay to pull up the poll? So we actually, we have a quick survey for you to do, if that's okay, just to take a minute or so. The idea is to gather the interest on campus for the CytAssist instrument. So as Leilani explained, it has a lot of enhancement for spatial transcriptomics. So if you mind, just there's just 4 questions for you to answer. Thank you so much.
Yeah, maybe while that poll is running, thank you everyone for participating. Thank you, Leilani, for the talk. So many cool and interesting and powerful tools and technologies that you guys have currently and are continuing to develop and evolve. I just want to remind this group of what we currently have. So we do run Visium, FFPE, Fresh Frozen, as a service in collaboration with the Imaging Center, our BLS Histology Core, as well as University Genomics Center. So we do have, that expertise front to end, especially on the histology side of things. So please do contact us for those projects. But please do reach out to us. Let us know about your future funding plans, your goals, if there's something that we can help with, because obviously there are a lot of exciting things going on. So please do share those things with us and reach out to us and see how we might help. It looks like, I think we'll give the poll a couple more minutes, but if there are questions, please do raise your hand, drop something in the chat, or just unmute yourself and go ahead and ask.
We'll wrap up the poll here, Grant, and just maybe a few more seconds here. It looks like the answers have mostly stopped coming off, and then we'll share the results with the group. So I believe everybody should be seeing these now, but a good mix. Most participants will be actually submitting samples soon, and then a good spread across when they'll be ready to submit, as well as number of samples and species they're studying.
Thank you so much, everyone. We're happy to take any question as well. And I did put my contact information in the chat. So if people would like any information, but also on the data set for Xenium, please feel free to contact me and I will send you that information directly.
Well, maybe I'll go ahead and start with a general question. A lot of awesome tools that really seem to complement each other. Where would you suggest somebody starts? In the middle with that kind of sort of exploratory, non-hypothesis driven approach? Where would you start, or how would you start to think about maybe planning an experiment?
So that's a fantastic question. I think the first question to ask is what tissue type or what species are you working with? If you're working with something other than human or mouse, then you have the ability to work with our direct placement fresh frozen technology. That's our species agnostic technology. So that's usually where I start people off. When working with human or mouse, you have a few different capabilities. So now we've added capabilities on our CytAssist instrument for both fresh frozen and FFPE. You can either do direct placement or CytAssist for both. And so at that point, it just depends on what samples you have access to. Do you have access to being able to section from that block directly? Do you have something that's already on a plain glass slide? And I think those are the questions that people have in terms of the biology. And all of this obviously would be for our discovery tool, which is Visium. For anyone that has questions about Xenium, at that point, it's compatible with either fresh frozen or FFPE, but it is going to be a much more focused approach. So if you have a discovery question, Xenium is not going to be your best option. You want to have kind of your targets in mind. And a lot of people, what they're doing is they're using either their single cell or their Visium data to actually inform what they're using in terms of Xenium probes.
Thank you. Oh, makes sense. Any other questions?
I have a question. First of all, thank you for the presentation. It's very, very informative. With regard to the protein panels that are going to become available, so the first one being the immune panel, that's very exciting. Are there more coming? I suppose so. And also, what's going to be, is there going to be the flexibility for folks to choose their own panels?
So that's a fantastic question, Fernanda. And yes, while the immune profiling panel is the first protein panel that we'll be adding to our CytAssist platform, we will be adding there will be additional panels that will be added. And then the other thing is that we've already talked about the ability to, even those original panels, you can spike in, so to speak, and add your own custom antibodies of interest. And that would be something that you would have the capability of without the panels as well. And so it's going to be very enabling, once again, for users to be able to use whatever tools of interest that they have. And so if they want to use specific antibodies that they may have already worked on in the laboratory, it's then a matter of either working with a company or themselves adding a barcode to it. The advantage of these panels that I should have mentioned earlier is that it's not immunofluorescence based, so it's going to be an additional sequencing readout. So you can really load up these panels and really get to high levels of multiplex information. So I mentioned that the immune profiling panel is 35 antibodies at once, for example.
That's interesting. It's going to be sequencing readout then. All right, I'll be interested. Well, I'm sure you guys will be able to share once you have the protocol and the downstream part of it. Cool.
Yes, our product management teams have promised that this is coming out towards the latter half, this latter first half of the year. So I'm really excited. I'm hoping that we begin to get this information and maybe even info for pre-orders soon. So
great, thank you.
Let's see something in the chat. So a question from Craig Burrow. There's been a lot of development in the last few years for your probe-based human or mouse type reagents. Is there anything in the pipeline that will update your species agnostic fresh frozen workflow? That's a fantastic question. We are currently working on developments on that side of things. One of the things that we are also enabling users is the ability to generate probes against other species. So I mentioned that we have some work currently looking at, for example, non-human primates with our human panels, and we have some users that are doing that. We're currently working on potentially developing panels for other species of interest. It's just going to be a matter of how many people are interested in a particular species. And then for our fresh frozen approach, I think it's just one of those things where it's been using that poly capture methodology really enabled users across the board. So I think that's kind of why we've stuck with that as an offering thus far. But we are always looking to advance our technologies, and I would not be surprised if we continue to do so within our product management team.
How about resolution for that fresh frozen agnostic one, not CytAssist, right? But because it's not an option.
I totally understand where that's coming from, and I know that that's actually a big question that people have, like whether we would be able to increase resolution. It's something I know that we've been working on internally. I think for the reason for pushing on the CytAssist is just because we've seen such enhanced data compatibility thus far. So we've seen that really increased specificity and sensitivity with our CytAssist instrument. And that's something that comes from two different aspects. One of them is using a probe-based approach, and so that really allows for higher sensitivity. You're working with much smaller sequences, and you can imagine as you're working through a tissue, for example, if you're trying to release RNA from a tissue onto these capture oligos that are underneath, how having, these long, potentially curving RNAs can, with secondary and tertiary structures, can potentially cause an effect as to the diffusion. And so probes are much smaller and they wouldn't have that problem. That's one of the aspects. The other thing is when you're doing your transfer with the cytosys, once again, you're not going through a complete tissue, you're just working with kind of that top level of the tissue overall. And as that happens, the diffusion is much easier, much quicker, which is one of the advantages of the cytosys platform. And so I think that's why we focus on that thus far. We've just seen that enhanced capability. I wouldn't be surprised if in the future we do come out with something. It's just going to be some level of technological limitation where, we've already seen this advancement at this level, and that's why we focus on it.
Thank you.
Of course.
Anybody else? Last call. Doesn't look like it. All right, we are at the end of the hour. We know how to get a hold of you, so if anything does come up, please get a hold of the spatial team or our 10X partners directly. That will do it for this month. Thank you very much to both of you. Wonderful talk, a lot of great things going on, and we will see everyone else on April 25th. Thank you.
Thank you, everyone. Have a great day.
Bye, everyone.
Bye. Thank you.